Bismillahirrahmanirrahim. Today we start with the transport in plants in AS level biology 9700. The second video in this chapter, which is going to talk about transpiration and translocation in detail, which we did not cover in the last video. Looking at the syllabus once again, we have finished part 7.1. Now today we're going to do 7.2, which is the transport mechanism. So please everybody refer to this once we have finished the chapter and we finish the videos on it. So the main ingredients are going to be about the different pathways of water, the how we study the rate of transpiration, and then what is translocation, which is all in these last three. And we've got to study xerophyte. We've got to study xerophyte leaf, the leaves of xerophytic plants. So what are xerophytes? Those which can withstand high temperatures and less water, supply less water in the soil, which is of course desert plants. First of all, I want you to get acquainted with the structures of the two main tissues. One is the xylem and the other is the phloem. Now this is a picture of the phloem, and we look at it, we've got something very, very typical. Number one, it's got these sieve plates. A sieve is like a strainer. In Urdu we say channi, which is, you strain tea. When you're pouring tea, you don't want the tea leaves in it. So these are the sieve plates, and these are the actually plasmodesmata. You see these are connections between one to the other. You see this connection between this and this. So these are called the sieve plates. And then the other thing which is very important is you have cells which are adjacent to it and which are sort of friends of the phloem or the sieve tube elements. Phloem is also called the sieve tube elements. And the friends of the sieve tube elements are companion cells. A very nice word, companion. Companion means somebody who's there for you all the time, a friend, a confidant. So the companion cell, then it has these plasmodesmata connecting it. And the companion cell is a normal cell, but it's got a lot of mitochondria in it because it's going to help in loading and unloading. So companion cell, a normal cell next to the sieve tube element. Now I'm going to show you a few more diagrams to build your image or the understanding of it. Another very nice diagram showing you the pores in the wall between the neighboring sieve tube cells connect the cytoplasm and allow substances to pass freely from cell to cell. And companion cells lie next to the sieve tubes and they carry out respiration, protein synthesis for the sieve tube cells. Now the basic understanding about the phloem is that, as you can see here, we say sieve element, or we say this is the companion cell. Now the important thing which you've got to understand is this. Sieve elements have no nucleus. They have no nucleus, no tonoplast. There is no such tonoplast. It's missing. And tonoplast, please remember, was the vacuole membrane. There is no nucleus, there is no tonoplast, and there are no ribosomes. The reason why is because there is no nucleus, so the DNA is in the nucleus where transcription can take place. So if there is no nucleus, there will be no transcription, no translation. So why would we need ribosomes? So please understand is that we do not need any. So no nucleus, no tonoplast, no ribosomes. No nucleus, no tonoplast, no ribosomes. Then what do we have? We have the sieve plates or the sieve pores. Please look at it carefully. Mitochondrion, yes, a few. Cellulose cell wall. And then you have the cell membrane, or called the plasma membrane, the cytoplasm. You have a little bit of cytoplasm on the sides. It's called peripheral cytoplasm. And you also have the endoplasmic reticulum. But when you look at the companion cell has everything. Cell wall, mitochondria, rough endoplasmic reticulum, cell membrane, plasmodesmata, vacuole, tonoplast, middle lamella, nucleus, and cytoplasm. So the companion cells, as compared to the sieve tube elements, are highly developed and have everything which a normal plant cell would have. So a companion cell as compared to a sieve element. So please pause here, make your notes and make a mental picture of this image. Now I want you to remember and learn, number one, how a section of the stem looks like. Now we're talking of the stem. And which stems do we eat? Like we have sugarcane. So the outermost layer is called the epidermis. Then we have the collenchyma and the parenchyma. So I always say remember it by some way. You've got to remember it. So epidermis, collenchyma and parenchyma, and this together is called the cortex. So the collenchyma is the outer layer, which is, because you've got to be picking this up when you're looking at a slide of it. And then we have the major is the parenchyma, and then the middle area, which could be hollow at times and could have cells as well, is called the pith. And then you have the vascular bundles on the periphery, and the vascular bundles have the xylem on the inside, and they have the phloem on the outside. So the xylem and the phloem, epidermis, collenchyma, parenchyma, cortex, that's something new, and the pith, which is the central region, and the vascular bundle. So please pause here, do a diagram, and remember it. Epidermis, collenchyma, parenchyma. Then we come to the transverse section of a root. And a root, usually to say, is you just cut a carrot, which is a root, and you look at it. And interesting is, again, what we see similarly to the stem is number one, epidermis. But here the epidermis has root hair cells, and you can see this root hair cell here. Now in the center you can see the xylem. So the xylem in the center, and then you've got the phloem on the sides. The root hair cell, and then important thing is this area is called the cortex. So epidermis first, then we have the cortex, and then we have a structure which is known as the endodermis, and then just inside, inner to the endodermis, is the pericycle. So how to remember it? Number one, epidermis, cortex, endodermis, and then pericycle, and then of course you have the xylem and the phloem. So in order to remember it, you need to remember it in some sequence. Epidermis, which is this area, the outer area, which is just a very thin line, and then you have the cortex, which is this made up of cells. I'm not saying it's just an empty area, it's made up of cells. And then you have the endodermis, which is marked in sort of a bluish color, endodermis. And then you have the pericycle, and then you have the xylem and the phloem. So pause, please draw, remember epidermis, cortex, endodermis, pericycle, and then the xylem and the phloem. Then I want to discuss the different pathways that we have, and the name is very typical that we talk about. We talk of APOPLAST pathway. APOPLAST pathway. And I usually ask you to remember it. A for apoplast, A for along the cell wall. Along the cell wall. But it's actually not, it is along the cell wall, but we do not say that. We say that in biology, we say through the cell wall. Why do we say through the cell wall? Because the cell wall acts like a absorbent paper and sort of absorbs everything. So through the cell wall. Now if you look at the apoplast pathway, now that has been labeled a blue shade, this one here, and you see there's a xylem vessel. As the water is being carried up, the apoplast pathway is through the cell walls, and that's why the apoplast pathway is blocked when it reaches the Casparian strip. And the Casparian strip is that layer of suberin which surrounds the endodermis cells. So we have the Casparian strip. The Casparian strip is the layer of suberin which is surrounding the cells of the endodermis. And then of course after the endodermis, you see we've got the pericycle. So we've got the apoplast pathway and the SYMPLAST pathway. And a way to remember the SYMPLAST pathway is, there's a Y in it. So cytoplasm to cytoplasm. cytoplasm to cytoplasm. So apoplast pathway along the cell wall or through the cell wall, and the SYMPLAST pathway is cytoplasm to cytoplasm. So one way to remember it is, there's a Y in it, so cytoplasm, there's a Y in cytoplasm as well. Another very good diagram to show you, number one, the apoplast pathway, and you can see it if you just pause this video here, how all this, the arrow is going through the cell wall. And then the SYMPLAST pathway is transmembrane pathways is, the cell enters the cell, then from one cell to the other enters the cytoplasm, and then enters the cytoplasm of another cell. And then you can see here, this is the epidermis, this is the cortex, we're talking of a root. And then you can see here what we have is the endodermis. And the endodermis has these strips of suberin around it. I'm just drawing it with a red marker. And this makes up the Casparian strip. So it goes all around the root. You can see it's going all around it. The endodermis is surrounding it. The Casparian strip is all around in all the cells of the endodermis. And then inside to that is the pericycle. Inside to that is the pericycle, the cells of the pericycle. Pericycle is made up of cells, a single layer of cells. And then of course you have the xylem and the phloem. So everybody must know how to label this. Root hair cell or the epidermis is on the outside. Then we have the cortex. Then we have the endodermis. Then we have the pericycle. And then we have the xylem and the phloem. So please pause, do a drawing, and please try to remember it, because you have to do a lot of remembering, rote learning. Another important pathway of water, just a slide on that, soil water enters the root through the epidermis. It appears that water then travels in both the cytoplasm of the root cells, called the SYMPLAST. That is, it crosses the plasma membrane or the cell membrane and then passes from cell to cell through the plasmodesmata. So it's basically the cytoplasm through which the water molecule is traveling. Then the other, it says, in the non-living parts of the root, called the apoplast, that is in the spaces between the cells and in the cell walls themselves. And this water has not crossed a cell membrane. Plasma membrane is cell membrane. Now, as you get a lot of questions in paper 3 and in your MCQs as well, I want you to look at a vascular bundle. And I want you to always be very clear how you're going to label it. So this part of it, this is the xylem. And then we have, this is the phloem. And in between is the cambium, which of course adds more cells as the plant grows. So phloem and xylem. Xylem has these because lignin is deposited in them, so it's got these very rigid hexagonal shape, very typical of xylem. And it's sort of got these corners, very sharp corners. And the reason why is, and you see these very dark lines about them, because they have got these lignin deposited in it. So you see the xylem as very differentiating and very clearly different than the phloem. Phloem hasn't got any of these very sharp edges because there's no lignin in the phloem. Then let's look at xerophytes. Xerophytes are these which have been adapted to survive long periods of drought and high temperatures. So these are the ones which are to be called desert plants, xerophytes. If you look at the features of xerophytes, number one, they have sunken stomatal pits. It's got succulent tissues, leaf are reduced to spines, they have very long roots, stem with a rounded shape, and reverse stomatal rhythm. Reverse stomatal rhythm means that during the day, in order to prevent loss of water by transpiration, they are going to remain closed. Normally, In the daylight they will open, because so as to facilitate photosynthesis. But in these plants we say reverse stomatal rhythm. Reverse stomatal rhythm means that they will close in the daylight and they will open up at night, so that then they have another way of photosynthesizing. But reverse stomatal rhythm means closing during the day when there is a lot of light, so photosynthesis will be slightly compromised. Then a very thick waxy cuticle, and then rolled leaves. Please do not say folded leaves, it's very wrong to use the word folded. And they've got hairs, which of course trap a layer of air, and that prevents loss of water being carried away by wind, so there no gradient exists, so there will be less transpiration because of the hairs. Here again we have some pictures of succulent xerophytes with fleshy leaves. Of course the fleshy leaves are there because they can store water for longer times, so some nice images. Then we come to some xerophyte adaptations and a summary of it. Number one, thick cuticle. How does that help? It stops uncontrolled evaporation through the leaf cells. Then small leaf surface area, less surface area for evaporation, and we've got the conifer and the needles and the cactus spines. Then low stomatal density means less number of stomata per unit area. So of course, if there's less stomatal density means less surface area for diffusion. Then sunken stomata. Sunken stomata means that the stomata have sort of sunk into a groove, and that of course helps, because it is going to, the water vapor is just going to, not going to escape, but is going to stay in that groove and will not form the gradient. So if there's more water vapors inside the leaf and more in the groove, so there's going to be no transpiration. Marram grass is an example of that. Then stomatal hairs, which are called trichomes. And these trichomes maintain the humid air around the stomata. So then rolled leaves maintain humid air around the stomata, and extensive roots maximum water uptake. Now some names that I want you to be familiar with, because we will be talking about some of these names in the paper 5 in A2 syllabus. So mesophytes, plants adapted to a habitat with adequate water. Xerophytes, dry habitat. Halophytes, salty habitat. Hydrophytes, plants adapted to a freshwater habitat. So very interesting phenomena. And if you have time and you have a spare time, please do study these, because these really help you to understand how beautiful nature is and how God has been kind to us and given us all these different types of plants which are adapted to living in different habitats. Just a quick recap of most of the xerophytic adaptations. Please pause this and just read through this page. Dry desert plants, some other modifications: leaves modified into spines or hairs to reduce water loss, shallow but extensive fibrous root system, the surface area reduced by producing a squat rounded shape, stem becomes the major photosynthetic organ and a reservoir for water shortage. This is another very interesting xerophyte. Is a marram grass. You find these on sand dunes, and the leaves are rolled up to trap the air inside. It has a very thick waxy cuticle which prevents the evaporation from the surface, which is cuticular transpiration. Then trapped air in the center with a high water potential. The hairs on the lower surface reduce the movement of air, and the stomata are in pits to trap the air with moisture close to the stomata. So very interesting plant and a very interesting phenomena to study. Hydrophyte adaptations is not so much in the syllabus here, but I just like to discuss it here because we don't do a lot of plants, other plant biology, but here I want to just discuss it. Wide flat leaves that spread across the surface of the water to capture as much light. Very thin or no waxy cuticle, because there's a lot of water around it. There is no need to conserve water. Then open stomata on the upper surface of the leaves, because you see from the air, and these leaves float on the surface of the water. So the stomata are on the upper surface and the guard cells are inactive. And because the water is going to support the leaves and the flower, so they do not need any strong supportive structure like the lignin and all in the phloem and a lot of xylem. And small roots, why does it need long roots? It is in water, and the water can diffuse directly into the stem and the leaves. So not much of an issue. Very interesting how that plant is adapted to its own habitat. This is another beautiful image of a xerophytic plant and a slide of it, and this come in one of your exams papers as well. Now you see, the interesting thing is that it's got a very thick cuticle. Now you can see the cuticle here. I am just drawing the line to show it, the very thick cuticle. And then you see the upper multiple layer of the epidermis. The epidermis is nearly 3 layered. And then you have, this is the palisade mesophyll. And then you have the spongy mesophyll. And then again you have a 3-layered epidermis, and then you have the lower epidermis. So upper epidermis, which I'm shading in black, and then you have this part is the cuticle. The part which I just shaded with the cuticle is the non-cellular layer on top. And then we have the 3-layered or 4-layered at times the upper epidermis, and then we have the palisade, and then we have the spongy mesophyll, and then again we have the lower epidermis is also multiple layered. You can see in here also this part here, and you can see this part here as well. Well, of course it's a section, so you don't see it very clearly. And then you can see these hairs. Now what is going to happen? What is going to happen is that when water vapors diffuse out, they're going to stay here. When they're going to stay here, there's water vapors inside and there's water vapors outside, so there's going to be no gradient. And the movement of water vapor is going to stop, because the water vapors that have diffused out of the stomata are staying there in this groove. And the guard cells are here. This is where the stomata is. Stomata is inside grooves or pits that we say. So please understand this. We have to be very clear on how we perceive these slides, because you can be asked to do a plan drawing of this as well, and a plan drawing would be very simple. What would it be? It would only be two lines here for the cuticle, then this line here for the upper epidermis, then this line here palisade, and then we have this line. We'd have a structure like this. So as I say, you need to just trace it out. So a plan drawing of this leaf of a xerophytic plant. You could get this in your paper 3, a plan drawing. Now let's look at the function of the xylem and the phloem. The xylem is usually only going to transport water and mineral ions, and there are no end walls, and it's only one way. One way means it's only from the soil enters the root hair cell, and then it goes up to the aerial parts of the plant. Now in the phloem, number one, they're organic molecules, and the organic molecules are sucrose and amino acids in solution. Sucrose and amino acids in solution. And the end walls have sieve plates, and of course they have two-way movement. Two-way movements means in some sieve tubes, not in the same one, but in some sieve tubes it's going to be in the upper, is going to be upwards, and in some other sieve tubes adjacent to it, it could be in this direction. Because you see, it is from source to sink, from the leaves to the roots, or maybe from a leaf to another, to a white flower on the top of the plant. So source to sink. So some phloem will be taking it upwards, and maybe some phloem tubes will be taking it downwards. So we're not saying in the same tube, but in the tubes adjacent to it. So in many of the tubes they'll be traveling up, and many of them they'll be traveling down. Now coming to the uptake of water by the root hair cells. Now you can see here the soil particles, and in between the soil particles is the water molecules. So we've got, now please understand this, this is the epidermis, root hair cells, then we have got the root cortex cells, and then this is the endodermis, and then this would be the pericycle, and then of course we've got the xylem vessel. So what is to happen first is that the water from the soil has to enter into the root hair cell, and then it can enter, some of it can enter even straight into the cytoplasm. And then it takes two pathways. Number one, if you can see here the line, apoplast pathway, and then the SYMPLAST pathway. So you can see the colors and you can see how it has been shown. So apoplast pathway enters the root hair, then some of the water goes through the cell wall, which will be the apoplast pathway, along the cell wall, apoplast, through the cell wall. And the SYMPLAST is from cytoplasm to cytoplasm. But you see here how as soon as it reaches the endodermis, the apoplast pathway is blocked. So the Casparian strip blocks the apoplast pathway. Blocks the apoplast pathway. So water entering the root hair cell, then going through the cells of the cortex, reaching here the endodermis. And as soon as it reaches the endodermis, the apoplast pathway is blocked, but the SYMPLAST pathway continues. And then of course it enters the pericycle, and then it enters the xylem. As soon as it enters the xylem, now there is going to be a suction force. And that suction force is the water is drawn up by the transpiration stream as a result of the cohesive forces between molecules. So cohesion is the hydrogen bonding between the water molecules. Adhesion is the water molecule adhering to the sides of the xylem vessels. Adhesion, cohesion. You must be very clear about this. So, and then of course, as it rises up, then it is going to move out of the xylem and enter the cells, the mesophyll cells, or any cell. And then that water vapor is going to escape through the open stomata. It will enter the airspace and then it will pass out of the stomata. So please remember, the water first has to evaporate. It has to evaporate at the surface of these cells. So first it has to be evaporation. So the water evaporates here, and then of course it moves out as a water vapor, not as a water liquid state, but as in a gaseous state, as a water vapor. The water vapors have to diffuse out. And please remember, this diffusion of water vapor is not going to be osmosis. Why not osmosis? Because there is no partially permeable membrane. It's just passing out of an opening. So please understand how the water has to first evaporate at the surface of the mesophyll cells, and then that water vapor in the air spaces has to diffuse out, and that is called transpiration. Transpiration is the movement of water vapors. Transpiration is the movement of water vapors out of the leaf through the stomata. First evaporation at the surface of the mesophyll cells, and then the movement of water vapors out of the leaf through the stomata. This completes the video 2 of this chapter, and this will be followed by the video 3, which will contain material on potometer and other parts of the syllabus which have still not been covered.